Welcome everyone, I'm Sephra Alexandra, lead of CT NOFA's Pollinator Health Initiative, the Ecotype project. Today, we are taking you on a virtual tour of our seed collector partners at Highstead. Let's go. Okay, so the important thing about the Ecotype project is it takes a lot of strategic partners. And for those of you that have now bought plants or have gotten involved with us, you know that first thing we need to do is actually source our seeds. So to do that, we want to make sure that we're sustainably wild collecting our Ecotype seed, and that is what we use to locally grow our founder plots that then grow plants that then you all can have. So ultimately what we're trying to do is have the right plants in the right place, and the beginning of that process starts with who I'm going to introduce you to today. Jordy Elkins and his co-seed collector Jesse Hubbard are going to talk you through the process of what it takes to identify a wild stand, monitor it, collect and clean the seed, stratify it, and propagate it, at which point it then goes to the next phase where we establish founder plots. So let's all go to beautiful Highstead and see what it means to be a seed collector.
You know, one thing that really amazes me about these meadows is just the diversity that's in them. You know, here at Highstead, we've got this incredible landscape with so many different types of plants. We have this common milkweed, which is common, obviously, but we've got at least 2 other species growing on the property, including purple milkweed, a plant of special concern in New England. But because we've preserved this land, it's really become sort of a place for species that really are of special concern. It's just really important to us to have a place here where nature can really thrive. But part of our mission is also to encourage others to adopt that same ethic. And so by providing plants to the nursery industry and to farmers who can then distribute them to homeowners who can turn their turf grass into a pollinator meadow, you know, that's something that's just really exciting to see, you know, sort of this landscape desert of suburbia really reclaimed with natural plantings that benefit pollinators and just the environment in general is really what our work is all about as far as land stewardship side of what we do here at Highstead. Highstead was started in 1982 by the Dudley family. They purchased the land and just absolutely loved it and wanted to do something where they could learn from it and where they could share it with other people. And so they started Highstead Arboretum as a native plant arboretum. Highstead started as an arboretum, but over time we've grown to be a land conservation organization, but our roots have always been here on this property, and we have a long history of working with plants, and that's what really led us to get involved with the Ecotype seed project. We felt that we wanted to do some restoration planting on this property, and we wanted to have seeds that came from the local area. And because of our connections with conservation organizations, we had availability of places to collect seed from that we wouldn't otherwise have opportunity or access to those properties. And so because of our plant science background and our connection to large nonprofit landowners of natural lands, it really put us in a good position to really lead the seed collecting part of this Ecotype project that we're involved with. Before we head out into the field, we do a lot of planning about what species we want to look for. We started with species that we knew were readily available in the nursery trade, because one aspect of this project that is important to us is to get plants that are available to homeowners and to get ones that they're familiar with, but that also benefit pollinators. We didn't want to find obscure plants that nobody was familiar with because we figured nobody would plant them. And so we began looking through nursery catalogs and cross-referencing what was available with plants that we also knew were well suited for pollinators. So once we decide what we're looking for, we go out into the meadows and we start scouting around places where we think we might find it. What is the right type of soil? What's the right type of ecology? What type of ecosystem does it grow in? And we do a lot of scouting in advance to look for it. And then the next thing we have to do is go out and identify the plant and make sure it's actually what we're looking for, because once it's in seed, it's really difficult to tell. So for instance, you know, this penstemon, we'll go out and hunt the fields and look for it, confirm the identification, and then mark where it's growing, because it's easy to see now that it's in flower, but once these flowers are off and you're in a large meadow full of hundreds of other different species of plants, it's hard to get back to. And so there's a lot of prep work that makes the seed collecting more efficient when we head back into the field. Before we actually do any seed collecting, we always get permission from the landowner. And so that's what happens, you know, once we identify a population that's suitable for collecting from, then we go and get written permission. One thing about finding the species and collecting the seeds is you need more than one plant. You can't just find a single plant growing in the wild and collect seed there. To really have a diverse gene pool in the collections, we need to find populations that have at least 50 individual plants. And while that doesn't sound like a whole lot, in some species it can be quite difficult to find that many plants growing together in one area. And then when we go out and collect, we want to have a really wide representation of all the genes that are in that population. So we don't just collect all the seeds from one or two plants, but we want to go and collect across the whole spectrum of all the plants in that population. All the while wanting to be very cautious that we're not threatening that wild population that's there by over-collecting. So we just take a very small percentage of the seeds, usually, you know, under 5%. We never take over 20% of any individual plant or any population in total. So we're really careful about the protocols that we use and wanting to maintain the viability of the populations that we're collecting from, making sure we're not damaging them. This is Jesse Hubbard, one of my co-workers, and he's a huge help in collecting the seed. It's really a big effort to go out in the field, and a lot more than one person can do. And so Jesse helps with a lot of that work.
Working with Jordy's been a great experience because I've learned a lot about, you know, all the native plants in our area, how to collect seeds responsibly, first identifying the right plants, collecting in a way that we're not harming the populations. It's an exciting thing for me to be out here and doing the collecting, to still be involved in the process through the stratification, the sowing, and seeing the end result of the plants that are growing from the seed we collect. So here are sieves that we use in cleaning our seed, and they're all different sizes. You know, so we're trying to separate the seed from the chaff or the shells. This is a number 14, and you know, the smaller the number, the bigger the holes. When seed cleaning, it's really just trying to come up with a protocol, I guess, for each different plant, because they all kind of have their own characteristics as far as how they separate from their shells and chaff and everything. So it's really just trying to find the best method for each species. You put it in the refrigerator throw a moist paper towel in there to kind of keep it, it's called a moist stratification. So, many of the seeds require about 30 days, but it varies depending on the species. So in stratifying the seeds, under moist conditions, it helps to break down the outer, like, hard shell, and kind of mimics what goes on in nature when they're sitting in the moist ground over the winter. It speeds up the germination process. These are Pycnantum Tenuifolium, very narrow-leafed mountain mint, and the seeds are tiny, as you can see. And it's tricky not to mix them with other seeds when you're working with them, but these are one of the smaller ones. And this is going to be the species that Jordy will be talking about.
Here's a tray of finished seed after it's been stratified and then sown and grown in our greenhouse. This is the final product, and so this is a 72 cell, and these are ready to plant outside. You can see how well rooted these are. And usually if you plant them by spring is ideal, but if you can get them in the ground by midsummer, they're usually fine to make it through and to be established before winter sets in. You know, the need to produce seed is beyond what we can do. It's really a collaborative project. And so we can't possibly grow all the plugs that are needed to really support the demand. But being a science organization, we like to understand how each of these plants grows and what the requirements are. And so we grow it here ourselves, and we really record, you know, what are the characteristics, what does it take to actually get it to grow, and then we can document that and share it with people that we distribute seed to. Giving the seed and giving the information to others who can then do some of the work really allows this project to scale up far beyond what we could do if we were the only ones working ourselves.
Thank you everyone for joining our virtual farm tour of Highstead. Hopefully now you all understand a bit more about the process of what it takes to do wild seed collection and make those plants propagated and ready for our founder plots so we can all get more of these ecotypes to you. Stay involved with us at the Ecotype project, and we hope to seed you soon.